first and foremost, I'd like to thank the organizers for giving me the opportunity to talk to you today. And yeah, today I'm going to talk about aerotactic waves in a living cell. And the title might sound a bit obscure, but essentially we are going to talk about an experiment where you have cell division that will combine with aerotaxis, and we will kind of question how those two ingredients combine together. Okay, and feel free, of course, to interrupt me if there's something that is not clear. So a little bit for the context and the motivation of the work. In nature, in real biological systems, cells are often exposed to enormous variations of oxygen. For example, in human tissue, you have variations from 1% to 20%, and this naturally raises the question: well, how do cells adapt to these variations? And so Mete and others in 2018 came up with a very nice experiment where they showed that cancer epithelial cells escape hypoxia through self-generated gradients. So we have talked a bit about self-generated gradients this morning, but I will explain it again later on. And so basically, so there's a team of biophysicists from Institut Light and Matter in Lyon, composed of Christophe Anjard, Olivier, Cochet-Escartin, and Jean-Paul Rieu, who basically adapted a similar experimental setup for Dictyostelium discoideum, so Dicty in short, which is an amoeba of size 50 micrometers that is swimming around, crawling around. And with Vincent Calvez, my PhD advisor, we basically contributed there to mathematically model the experiment and yeah, contribute like this. Okay. So the experiment is actually very simple. You take a population of Dicty, about 2,000 cells, and you confine them vertically between two plates that are very narrowly spaced. And so if you take a transverse cut of what's going on, what you see here is basically a dense cellular spot. And at time t equals 0, you have an oxygen, you have a homogeneous oxygen condition, but the cells will consume very fast oxygen, will start consuming the oxygen very fast, and basically the profile will go down. And what we will see is essentially you will have the emergence of a gradient here, and cells have Dicty has the ability to do aerotaxis. So that means that it can move from low oxygen regions to high oxygen regions. So this is exactly what they will be doing. So they will essentially start moving outwards. And so what you see from above is essentially the emergence of a ring that will expand, and it expands with constant speed and with a profile that is very that is also constant in the traveling frame of reference. But interestingly, the movement is sustained because of the fact that cells will move outwards, and then they will continue consuming the oxygen, and so this will further push the oxygen gradient away. So what is actually happening is that the cells are creating themselves the oxygen gradient, which is the directional cue. So they are generating themselves the directional cue, and that is what we call this framework of self-generated gradients. Okay. Oh, and here we have some photos. So here we have this time t equals 0, we have this dense cellular spot, and very soon afterwards you see the emergence of a ring, and then for a time later on you change the microscope, and you have this really nice ring that is very symmetrical, and also the width of the ring is very very narrow and stays constant over time. Okay. So we've been talking about when we started working on this project, we were talking with the biophysicists, and they were telling us that for them only two ingredients intervene in this. So the first one is aerotaxis, so this capacity to move from low oxygen regions to high oxygen regions, and basically in the ring, in the expanding ring, the cells will undergo aerotaxis. But then the cells that are left behind, they will not be able to do aerotaxis, and the reason for this is that the gradient becomes too shallow, so they cannot integrate the information where they should direct. And quite to the contrary, the cells that are outside of the ring, they will not do aerotaxis neither, not because they can sense the gradient, but because there's no incentive for them to augment their surrounding oxygen levels, right? And then the secondary ingredient is basically cell division, and it works very easily. Essentially, if you have enough oxygen and cells, their metabolism will work fine, and they will divide. And if not, then they will stop dividing. Okay. And so I mean, this is a bit a schematic view, but exactly where those transition happens and so on, that's not completely clear because I mean, the biophysicists have some idea, but experimentally, it's not so easy to exactly determine the behavior at each spatial position, okay? So the first question we want to ask is essentially, we have this traveling wave, and how can we explain the emergence of this traveling wave? Are the ingredients, aerotaxis and cell division, enough to explain it, and what's exactly going on? And the second one is based on the observation that in this experiment, we have, in fact, two different collective propagation phenomena. The first one is aerotaxis that I have mentioned, and the second one is due to the fact that we are adding cell division. And as most of you probably know, if you take a cell population that divides and at the same time diffuses, then, for example, by the description of Fischer-PP equation, you will actually have an expansion of the cell population, which will go at a constant speed to square root of RD, where R is the growth rate and D is the diffusion constant, right? So since there is cell division, there must also be somehow a little bit of this, okay? And the question that we are asking is, well, how do these two phenomena combine, right? So as Chris has talked a little bit about go, work, go, I'm going to approach it more from a modeling point of view. But essentially, when we started the collaboration, we wanted a bit to understand better how these two phenomena combine. And so we came up with this very minimal model. And I stress it, the aim is not to completely describe what is going on, but it is rather not a toy model, I don't like the word toy model, but more a minimal model that that we hope shows that the ingredients that we are using are enough to explain propagation under the waveform. And maybe this model will also give us insight into more complex models. Okay. And so the hypothesis is essentially very, very simple. The cells will have two distinct behaviors. If the surrounding oxygen level is below a certain threshold, then they will do aerotaxis and move the oxygen gradient upwards, but they will not divide because they don't have enough oxygen. And if the oxygen level is above a certain threshold, then they will so what we call grow, so they will divide, but not have a bias motion. Okay. So to fix the ideas, what we will have is to the left we will have this go, so this drift towards the left because the gradient is oriented like this, and to the right we will have this grow phase where we will have the division. And so if you are saying, well, that sounds familiar to me, it is because it is very similar to what Chris has been talking. So in fact, for example, Burger's Fischer KPP is essentially the same thing. You will have a drift to the left and you will have a growth to the right. So the transition is not exactly the same as Burger's Fischer KPP, but in spirit it's very similar, and that's also why the two models are in fact related. Okay, so what we will have is essentially a system of PD where the cells undergo the diffusion here, and they move with speed A0 if they are below the oxygen threshold in the direction of the oxygen gradient. And if they are above the oxygen threshold, then they will divide with rate R0. And we combine this with a dynamic, with an equation on C that models consumption by oxygen. And very interesting, this equation is, so it's very simple, and it becomes very interesting because the coefficients of the equation are piecewise constant. And this will enable us to compute explicitly a lot of stuff. Yeah, and also the coupling here in this equation really goes through actually the position of the threshold. So if you know where x bar is, then you can say, well, to the left you are going, and to the right you are going. Okay. And so the first question is, well, does this model exhibit a traveling wave solution? And the answer in short is yes. So there's a minimal velocity, and for every velocity larger than this velocity, you have a traveling wave. And we have an explicit formula for the minimal velocity. And there's essentially a dichotomy between what we call the large aerotactic bias case. So if A0, the advection speed, is larger than this threshold value, then you have that sigma star, the wave speed, is a combination of A0, R0 times D. So you have somehow a non-trivial combination between aerotaxis and cell division. And in fact, this is exactly in scale version, this is exactly what Chris has been shown. So if you take A0 equals high and R0 times D equals 1, you get this chi plus 1 chi. So it's exactly the same thing, but I wanted to keep the units. And in the smaller aerotactic bias, so if elevation speed is not high enough, you will essentially go with Fisher KPV speed. Okay. So this model is very nice because we can compute stuff, but it does come at a cost, which is that the existence and the uniqueness of the PD system was not so easy. So I'm not going to go too much into details, but it was quite a hard problem, and we managed to deal with it in the framework of monotone oxygen profile. And in the case of the monotone oxygen profile, what we have is essentially that we will have a unique curve, we will have a unique threshold for each time. And so we are essentially interested in the study of the dynamics of this threshold curve, so this XRT. So essentially, instead of dealing with the PD, we will be dealing with the study of the dynamics of this one-dimensional curve. So of course, the PD will be insightful for this and so on, but then we will apply a fixed point theorem. And so I'm not going to go into the details, but you have to, in order for the stuff to work, so the dynamics of the curve is given by an ODE. And in order to give sense to the ODE, you have to do enough regularity estimates which are not quite trivial. So there's a little bit work involved. But at the end, we show existence and uniqueness local in time. Okay, so next point is, in my results, I said, well, you have a minimal velocity and you have other velocities. But in fact, the question is, the minimal velocity should be interpreted as the biological relevant condition, speed, sorry. And so Chris has been showing a lot of results on the asymptotics of the level sets and so on. So we would be, you would say, well, the result should rather be easy, as I hope he will have convinced you. But in our case, we don't have a comparison principle. So the fact that we are lacking a comparison principle makes it much more harder. And so we just have a result where we show that essentially the propagation speed, the limit sub must be above the minimal velocity and the limit inf must be below the velocity. And we are not able to do better, essentially. Okay. So next point is we have been talking a lot about push and pull waves. So I hope that now you are a little bit, you know what the difference between a push and pull wave is. And if not, then I will have some graphic illustration for this. So Florian has talked about neutral refractions, I think, yesterday. So essentially what you are doing is you will label your traveling wave, subpopulations of your traveling wave with markers that do not interfere with the dynamics. So it's as if they were not there. And you let evolve the traveling wave and you see afterwards, well, how did the cells mix? And so if you take the right-hand side, you are in the small aerotactic bias case, so when the speed is Fisher, KPP speed. And what you will have here is then, if you take the turquoise cells, Well, after some time, actually, the front will be composed almost exclusively of turquoise waves, and of turquoise cells, sorry. And so what this means is essentially what it is the turquoise cells that are propagating the wave. And so that this is what we call by pull. It is really the cells at the leading edge that will pull the wave and that will make the wave propagate. Okay. And on the left-hand side, it is we have this. So it's the other case where the aerotactic bias is larger, and there it is quite different because cells from, for example, from the back will end up in the front, and cells from the front will end up at the back. So there's a mixing. Okay. And so this is, I mean, this is exactly this pull and push case. And just one explanation. So in the pull case, it explains also why A0 does not intervene in the traveling wave formula, speed formula, because essentially it's the cells that are at the leading edge that propagate the wave, but the cells that are at the leading edge, I should point this direction, but the cells that are at the leading edge, they will never sense aerotaxis. They will always be in the grow phase. So in fact, that's the reason why you should not expect having A0 that intervenes in the waveform. Just how much time do I have left? 10 minutes. 10 minutes. Okay, perfect. So the second question is. Not the second question. So another question is, so we have this nice model and so on. We have a wave speed formula and stuff like this. We can do a lot of analysis with it. But the question is a little bit, what is this go model in fact relevant? And in order to test the relevancy of, I'm not sure it's the word, but in order to test this hypothesis, what we did is we introduced a second threshold below which aerotaxis will stop. And what we then observe is that, in fact, profiles, the profiles that we observe are much closer to what we expect, what we see in the laboratory. But very interestingly, the speed remains close to the wave speed formula that we have found. This can be checked numerically and analytically. But there's a reason why the wave speed should be close to it. It is essentially because where does the dynamic between those two models diverge? It diverges at the back. So in the single threshold model, you still have, even at plus infinity, you will have waves cells that are pushing. But those cells are pushing, but they will not really contribute to the propagation of the wave. So they will just be left behind. So the fact that they are pushing or that they are not pushing essentially does not should not contribute substantially to the propagation. Exactly. And well, with this model we can show similar properties and stuff like this. And then afterwards we can actually investigate also more complex models. So that's what we did also with the biophysicist team. And from but we do this numerically, so the analysis becomes much more involved. So the question I still want to deal a little bit with is essentially, if we have more complex cellular response functions, do we have the existence of a traveling wave? And the second question, which is a little bit related to it, is so as I said, you have a minimal velocity and you are essentially selecting the minimal velocity. But this selection phenomenon of so which traveling wave you are really selecting from, that's really a question I think would be very interesting for this model, and I have no idea for now how this will turn out. But I think it's a very interesting question. And so almost to conclude, so with Milica Tomasevic and Vincent Calvez, we came up with a stochastic individual-based model. I'm not going to talk about it, but in order for the stochastic individual-based model to make sense, we came up with a new go-or-grow rule. And essentially what we are saying is, well, if I have, so forget about oxygen. If I have enough cells to my left, or I mean, if I have enough cells to my right, then that means that the cells that are to my right have, in a sense, consumed all the oxygen, and that means that there's no oxygen left. So instead of considering an oxygen field, essentially we will just define the threshold position with this condition that to my right I must have exactly mass 1. Okay, and so this will set the dynamics of the. So this will set the threshold between the go or go. Okay, and so this leads to the following PD, and it is much easier to analyze. So one reason. So just one instance, for example, in the chi bigger than, so here I have normalized variables. So I forget about A0, I forget about R and D. R and D is equal to 1 and A0 is equal to chi. So I come back to the notations of Chris essentially. And so in that case, for example, you can show much easier asymptotic stability when chi is bigger than 1. And although we've shown it in that case, transposing this to the case where we have an oxygen field, it's much more complicated. Okay, and so in fact, so, just the last slide is essentially about questions. So remember, we wanted to know whether the traveling wave speed was the Cauchy problem was converging to the traveling wave, and if we have convergence in speed, for example. So we had this result of lim inf and lim sup, and because the model here is easy, analytically better tractable, essentially what we expect is the asymptotics that Chris has been writing down for Fisher KPP, but which are the same here. So in the chi strictly bigger than 1 case, you have this convergence. So you expect to have convergence in sigma t plus O1, so no Brahmsen shift. If chi is strictly less than 1, you will have this standard Brahmsen shift of -3 halves. And in the chi equals 1 case, you expect to have this -1/2 of log of t. So this is still under investigation, stuff like this, but we have some, we are getting there slowly. Okay. And as Chris has pointed out, actually this is quite related to the Burgers-Fisher KPP equation, but also this reaction diffusion equation where you have this monostable cubic form, and also this free boundary problem proposed by Julien Belsiki, Rida, and Brunet. So yeah, I think what I'm very excited about is this idea of having a general framework to treat all these questions under the same, in the same framework, right? And as a conclusion, so what we've shown is essentially we came up with this minimal go-go model, and it proves that aerotaxis, subgenerated gradients, and cell division are enough to trigger a traveling wave. We came up with this explicit formula. We've shown that there's this dichotomy, and this dichotomy also translates into a dichotomy on pushed or pulled waves, and that to some extent these results can be generalized to more involved models. And finally, so as a cherry on top, I just want to point out that I think that these self-generated gradient models are really, really interesting from a biological aspect. And so there's a team around 3D that came up with an experiment that I want to show, and then I have finished. Everyone sees. So the first part is a computer simulation, but later on you will see the same thing under an experiment. And what they do is they take a maze. So the left maze is actually a little bit easier because the dead ends are shorter than the right maze where the dead ends are longer. But they will take Dicty cells will go inside, and so the heat map represents the oxygen concentration. And so what is very interesting is that the cell population, in fact, really, I mean, it flows almost amazingly through the maze by really finding the way, and it's essentially the oxygen gradient that are generated by cells, which give them the directional cues. Okay, and so this also works in vitro, where you can really see how those cells move through the maze, which I think is very interesting. So that's also a little bit the reason why we are so much interested in self-generated gradients and stuff like this. And that's it. I close this. And I'd like to finally thank my collaborators, so Olivier, Cochet-Escartin, Jean-Paul Rieu, Christophe Anjard, sorry, Melissa Tomasevic, Vincent Calvez, and of course, may I say my fellow Tussaudian Chris Henderson. And thank you for your attention.
Okay, so I have a bit technical question. Yeah. How do you deal with the discontinuity in the advection term precisely?
Basically, I just work in a framework where the threshold is exactly defined. So it's, I mean, I cross the threshold and I know it's at this position x bar t, and then afterwards I put myself in the frame of reference where this is centered, and then I factor out I somehow factor out the advection terms why I multiply with exponential times the integral of this thing, which makes it so it enables me to plug out from the conservative form to essentially pass from the conservative form to the non-conservative form, and then afterwards it's easier to deal with. Yeah.
I had a question about the simulations at the end. So not the in vitro, but the other one. Are you using the PD there? Or are you using an individually small, or what's the
very last ones?
Yeah, not the one with the actual Dicty, but the one with the heat map and whatnot.
Yeah, so the ones from 3D, like the video I showed.
Exactly. Yeah. So were they using the PE model, or were they using?
No, yeah. So we're using a singular, individual-based model.
Have you tried using your PDE model at all for this?
I have not. I think it would be very reasonable to check. Yeah. But I would not be surprised that it works essentially in the same fashion. But yeah, it would be nice to check it out. Yeah.
Okay, thanks very much for this talk. So I had a question about the results that you expected for your front position. So you said that the front should be trapped between let's say the linear expansion plus the log correction, and then you said okay. There's an off-one correction. So do you expect your wave to be unstable of some sort, or?
So we're talking here, and
yes.
Okay, so that's yeah. The question of stability is so in the case of the in the push case, it is very clear that it should be. I mean, yeah, we actually showed off the words. So, but what happens exactly in the pull case? It is not so clear for me. I think there have been works in the 80s on reaction diffusion equation, but there are probably many people that are much more aware of this, where they somehow, I think, where they do have, if you take the, no, I don't want to say anything wrong, so I'm going to retract, but I think in a general sense, it's I think we should make clear what asymptotic stability means there, and maybe in a specific framework you can get somehow something that is close to asymptotic stability, but I'm not sure about this.
No, it was just a small question. In fact, I just noticed that you put this off-one and not a constant plus something smaller. So I was just wondering if you expected some oscillations or something like that.
So this expected results actually. So there's an argument in Julien Vestiqui's paper with Brunet and Derrida, where they have this formal computation. And from the wave profile, from the explicit wave profile, you can exactly get the asymptotics. So it's completely formal, and it's I don't see how you do the computations rigorously, but it really gives you, it should really give you the plus constant plus even the lower order terms afterwards. Yeah.
Okay, thank you, Mete.